Hi class, in this recording we're going to dive in and start talking about some more general features of the brain, including the meninges and the ventricles. So as we look at the meninges are really the membranes that help to protect the brain. These membranes or layers that wrap around the brain are threefold. So as we look at the meninges, these bands of, there we go, now I've got my pen selected. These bands of connective tissue that make up the meninges. Little slip there. There we go. Meninges envelop the brain. And there's three bands, the dura mater, the arachnoid mater, and the pia mater, from superficial to deep. So dura mater is on the outside, arachnoid mater is in the middle, and the pia mater is the deep between the three. And these meninges are going to help separate the nervous tissue, the brain, from the underlying bone. The meninges of the brain are analogous to the meninges of the spinal cord. So when we think of the meninges, really it's three membranes that separate the central nervous system from the outside of the body. As we look at the meninges, they help to provide structural framework for the arteries and veins that help to supply nutrient delivery to the brain. And if we have an inflammation of the meninges, that is known as meningitis. Itis is a prefix we haven't talked about a lot in this class. Excuse me, a suffix we haven't talked about a lot in this class. The suffix itis means inflammation. So meningitis is inflammation of the meninges. It's a pretty serious disease in infancy and childhood. Individuals between the ages of 3 months and 2 years are at elevated risk of developing meningitis. Meningitis is typically caused when there's a bacterial or viral infection of the central nervous system, and that bacterial or viral infection of the central nervous system usually gets in through the nose or the throat, in particular with the nose, with the olfactory bulb and cranial nerve number 1. There's going to be a lot of neurons being exposed to the external environment, particularly the dendrites of those neurons being exposed to the external environment. And that is a frequent method for those pathogens to access the central nervous system. So as we're looking at the dura mater, I love the dura mater. Dura mater is German for tough mother. Dura meaning tough, mater meaning mother. And as we look at the dura mater, there's two layers to the dura mater. We have the outer layer, which is the outer periosteal layer. Peri meaning perimeter, around the perimeter. Oste meaning bone. The outer periosteal layer is going to go directly against the bones of the cranium. Here is some of those bones right now shown in the figure. We also have the inner meningeal layer. This inner meningeal layer is, well, as its name implies, going to be pressed against the other meninges. So we have the outer layer, the outer periosteal layer, the inner meningeal layer. And those inner and outer layers of the dura mater are going to have dural sinuses in between them. These dural sinuses that are in between them, I should shift to bright green here, are going to be reservoirs or pools that help to collect blood that's circulating through the brain. These dural sinuses also serve as a pressure release valve. As we look at the dural sinuses, you can see how there's an extension of the arachnoid mater going up into the dural sinus. This extension of the arachnoid mater going up into the dural sinus helps to regulate pressure within the brain, particularly pressure from the cerebral spinal fluid. It's worth emphasizing that within the cranial vault, there is no epidural space. This is a key difference between the brain and the spinal cord. In the spinal cord, there is an epidural space, so you can inject a localized painkiller and numb the spinal cord from a specific region down. We also need to emphasize that the dura mater is not attached to the cranial vault except around the foramen magnum, the sella turcica, crista galli, and then the sutures of the skull. And as we look at our dura mater, that dura mater is also going to invaginate into the brain. So here we can see the dura mater folding down into what is the longitudinal fissure of the brain, separating the left and right hemispheres of the cerebrum. Now, as we look at the arachnoid mater and the pia mater are in the brain and the spinal cord are going to have very, very few differences. So we're going to cover them both on the same slide. The arachnoid mater is stringy. It has a filamentous appearance. It looks like a spider web. So it's the spider matter in German. Arachnoid for spider, mater for mother, so the spider mother. And as we look at the arachnoid matter, it's pretty transparent, semi-transparent. If you ever had the pleasure, the joy, the privilege of doing a brain dissection, you'll find that as you peel the dura mater away from the brain, there's almost a slight tearing sensation as the arachnoid mater is moved away. Deep to the arachnoid mater, we have the subarachnoid space. And that subarachnoid space is going to have all of the filaments of the arachnoid matter. I need to back up a slide here. So I'm going to go ahead and right click. Let's go to previous. So as we look at that arachnoid matter, the subarachnoid space has those filamentous fibers, its namesake. Superficial to the arachnoid matter, let me shift to yellow here, we have the subdural space, the space deep to the dura mater. If somebody has a subdural hematoma, which is a fairly common injury that occurs, that's going to be blood pooling in the brain deep to the inner meningeal layer of the dura mater, but superior to, or superficial to, I should say, superficial to the arachnoid mater. Now, as we're looking at the pia mater, the pia mater is going to be very, very thin. During brain dissections, the pia mater is almost never removed from the underlying gray matter or nervous tissue. It's so thin, it's so filamentous that it's not really worth removing. It takes quite a bit of time. It's going to follow all the contours of the brain. It's going to go above the gyri, dip down into the sulci, and to truly view the pia mater and appreciate it, you typically need to look at it underneath the microscope. Now, as we're looking at the brain, our brain has a separate circulatory system from the cardiovascular system. This separate circulatory system is going to move cerebral spinal fluid around within the brain. And to help move and generate cerebral spinal fluid, we have hollow cavities. The hollow cavities in the brain are called ventricles. And there are 4 total cavities or chambers in the brain, 4 total ventricles. We have the 2 lateral ventricles. So we have the 2 laterals, which I'm highlighting green right here. You can think of the lateral ventricles as the left and the right, because we have an anterior view right here. And then after those 2 lateral ventricles, we have the 3rd ventricle, and then finally the 4th ventricle. And as we're looking at these ventricles, they're going to be connected to each other with some small tubes that help to move cerebral spinal fluid from point A to point B. Notably, in lab, we spend a fair amount of time talking about this tube right here that I'm highlighting as red, the cerebral aqueduct, which connects the 3rd ventricle to the 4th ventricle. Oh, there we go. So as we're looking at the 4th ventricle, that 4th ventricle is going to be located down here. It's going to be just superior, or just rostral, where the cerebellum connects to the midbrain. And as we're looking at our brainstem, and as we're looking at that 4th ventricle, it's the last stopping point of cerebral spinal fluid, or last congregation point of cerebral spinal fluid, the major one at least, before it is transmitted down through the central canal to the spinal cord. To produce the cerebral spinal fluid, we have a lot of choroid plexes. And as we look at the choroid plexus, or plexi, or plexuses, I'm not a very good grammar person when it comes to this. And yes, I did look it up, and plexuses versus plexi is used equally depending on how you taught your grammar. As we look at the choroid plexus, it's going to be a mass of blood capillaries and ependymal cells also. So here's the ependymal cells that are going to be located in the ventricles. The lateral ventricles are the 2 largest ventricles. So there's lateral ventricles. We have one there. There's another one right there. And they are going to have the largest masses of choroid plexus to produce most of our cerebral spinal fluid. So when we think of a choroid plexus, think the thing that makes cerebral spinal fluid. Because it needs to generate lots of fluid, it's going to have lots of blood capillaries and the ependymal cells present to make that cerebral spinal fluid. Now, as we're looking at cerebral spinal fluid, it's clear, it's colorless, and it's going to bathe the external surface of the brain. As we look at the amount of cerebral spinal fluid that's made, it's about half a liter, or 500 milliliters that are being produced per day. At any given point in time, we're going to have about 1/10 of a liter of cerebral spinal fluid in the brain. As we look at where the cerebral spinal fluid is made, it's 40% of it is going to be made in the subarachnoid space, external to the brain. 30% of it is going to be made as we look at the ependymal cells that just line all the brain ventricles. And then another 30% of the cerebral spinal fluid is going to be made by the choroid plexuses, which have a concentration of ependymal cells and blood capillaries. Now, I don't necessarily want you to focus on the specific ratios or percentages. Just know that, generally speaking, cerebral spinal fluid is made in many different parts of the brain where we have membranes or the meninges or the ependymal cells, to be more precise, in the brain. So really fixate on the ependymal cells making the cerebral spinal fluid. As we look at the cerebral spinal fluid, we're going to have blood plasma, and that blood plasma will be filtered. And as it's being filtered, it's modified by the ependymal cells to become cerebral spinal fluid. What do the ependymal cells do? Well, they add more sodium and chloride ions, and they remove potassium, calcium, and glucose, and make it so that there's very little protein present within the cerebral spinal fluid. So as we look at the cerebral spinal fluid, what's its function? First of all, it allows us to have buoyancy. Our brain needs to be cushioned and supported. And it's easier on our brain if it floats in cerebral spinal fluid. So as we look at the cerebral spinal fluid, it helps to buoy up the brain. If somebody has a spinal tap, a lot of times they get a terrible headache. For those of you who don't know, a spinal tap is when a needle is inserted into the lower portion of the spinal cord and cerebral spinal fluid is removed from the central nervous system. Removing that cerebral spinal fluid from the central nervous system makes it so that the brain sits lower within the cranial vault, and as that brain loses buoyancy because there's less cerebral spinal fluid to float in, many people will experience a horrific headache as their brain is resting on the floor of their cranium. The cerebral spinal fluid serves for protection. It allows the brain to be cushioned against impacts. If we look at somebody who has shaken child syndrome or concussions, these are going to be situations where even though there was some cerebral spinal fluid, there wasn't enough present, or the impact was so hard that there still was damage to the brain tissue. And then finally, the cerebral spinal fluid provides for chemical stability. We need to remove metabolic waste from the central nervous system, and then we also are going to need to provide our brain with new resources, new nutrients to generate more ATP. So as we look at cerebral spinal fluid and its flow, we're generally speaking, with all fluids, and yes, water is a fluid, air is a fluid, anything that can flow is a fluid. Cerebral spinal fluid is going to flow from areas of high pressure to areas of low pressure. So high to low. And most pressure for the cerebral spinal fluid is going to be generated around these choroid plexi or plexuses, because in terms of volume of the choroid plexes, volume per volume, or gram for gram, or the choroid plexes produce the most cerebral spinal fluid. So our cerebral spinal fluid, let me shift to green here, is going to start in the lateral ventricles, where we have the most choroid plexuses, and we'll continue down to the as we're moving, and we'll go to the third ventricle. From the third ventricle, cerebral spinal fluid will travel to the fourth ventricle. From the fourth ventricle, there's two paths it could take. It can go through the median aperture to the surface of the brain, or it can it can go to the surface of the brain, or it can go through the central canal down through the spinal cord. Now, we're constantly generating more cerebral spinal fluid, constantly pumping out more cerebral spinal fluid. Where does it go? If we're constantly making new cerebral spinal fluid, we need to constantly dispose of cerebral spinal fluid. Let's look at the end. We have in the sinuses, in between the outer and inner layers of the dura mater, we have the pressure relief valve that I mentioned earlier. These pressure relief valves are invaginations of arachnoid matter, referred to as the arachnoid villi. Let me shift to red here so it's easier to see. So here is an arachnoid villus. It is a protrusion, and a villi or a papilla is another term that could maybe describe this. It's a big invagination of arachnoid mater into a dural sinus so that we can have extra or excess cerebral spinal fluid be dumped back into the cardiovascular system and carried away. That's all we have for this recording class. If you have any questions, please feel free to post them on the class discussion board. Shoot me an email, and as always, happy studies.